Unit 9 Plant Biotechnology, which is an important unit for GAT-B exam. In this unit, we study how plant cells and tissues can be used in laboratory conditions to produce whole plants and useful products. It combines concepts from genetics, molecular biology, and cell biology. This unit is very scoring because many questions are direct and concept based. If you understand basics idea like totipotency, regeneration, and culture techniques, you can easily solve MCQs. So focus on clarity of concepts rather than memorizing too much. This slide mainly gives information about preparation strategy and support. It tells that concept-based learning is important along with solving MCQs and previous year questions. For GAT-B, practice is very important because questions are mostly application based. You should follow a structured approach. First understand theory, then solve MCQs, and finally revise regularly. Also, mock tests help to improve speed and accuracy. So, along with theory, always keep practicing questions. This slide gives overview of all topics in this unit planned by technology like totipotency, regeneration, PGRs, tissue culture, growth kinetics, and secondary metabolism. These are the core areas from which most GAT-B questions are asked. While studying, try to connect these topics. For example, totipotency is the base, regeneration uses it, and tissue culture is the method. Growth kinetics and metabolic protection are advanced application. So think of this as a flow, not separate topics. Totipotency means the ability of a single plant cell to form a complete plant. This concept was given by Hamberland. It is based on the idea that every plant cell has the full genetic material, but different genes are activated or deactivated depending on conditions. In tissue culture, we use this property by giving proper nutrients and plant growth regulators. Then the cell starts dividing and forms callus or embryos. One important exam point is that plants are mostly totipotent, while animals are not. Also, do not confuse totipotency with pluripotency. Regeneration means formation of a whole plant from a cell or tissue. There are two main pathways, organogenesis and somatic embryogenesis. In organogenesis, shoots and roots are formed separately, and this depends on the ratio of auxin and cytokinin. In somatic embryos, a somatic cell directly forms an embryo-like structure which can develop into a plant. Also, regeneration can be direct or indirect. Direct is without callus and is more stable, while indirect involves callus and may cause variation, which is important for exam. Plant growth regulators control growth and development in tissue culture. Auxins help in root formation and callus induction, while cytokinins promote shoot formation. The balance between auxin and cytokinin is very important for deciding plant development. Other hormones like gibberellins help in stem elongation. ABA is related to stress and dormancy, and ethylene causes aging. For GAT-B, remember examples like 2,4-D, auxin, and BAP, cytokinin, and especially the auxin-cytokinin ratio concept. Elicitors are substances that increase the production of secondary metabolites in plants. They act like stress signals and activate defense pathways in plant cells. This leads to increased production of compounds like alkaloids and phenolics. There are two types: biotic from organisms like fungi and bacteria, and abiotic like UV light or heavy metals. Important examples include jasmonic acid and salicylic acid. In exams, remember that elicitors are mainly used to enhance metabolic protection. Tissue culture involves growing plant cells or tissues in sterile conditions. The process starts with selecting an explant, then sterilizing it and placing it in a nutrient medium. After growing, subculturing is done, and finally plants are hardened before transferred to soil. The most common used medium is Murashik and Shu MS medium. Important conditions include temperature around 25 degrees Celsius, pH around 5.8, and controlled light. These are basic conditions frequently asked in exams. Cell suspension culture is prepared by transferring callus into liquid medium and keeping it under shaking condition. This helps cells to remain separate and grow uniformly. It is mainly used for large-scale production. There are two types: batch and continuous culture. These cultures grow faster and are more uniform compared to solid media. They are especially useful for producing secondary metabolites in industries. Growth in suspension culture follows a typical curve with four phases: lag, log, stationary, and decline. In lag phase, cells adjust. In log phase, rapid growth happens. In stationary phase, growth stops but metabolic production increases. For exams, remember that secondary metabolites are mostly produced in the stationary phase. Also, terms like specific growth rate and doubling time are important and can be asked directly in MCQs. Nutrient optimization means adjusting the culture medium in such a way that plant cells grow well and produce maximum metabolites. The most important factors are carbon source, nitrogen source, and pH. Usually, sucrose is used as the carbon source, and a balance of ammonium and nitrate is required for proper growth. The pH of the medium is generally kept around 5.6 to 5.8. If conditions are not optimal, growth and metabolic production both will decrease. For experiments, methods like response surface methodology (RSM) and factorial design are used to find the best combination of nutrition. Secondary metabolites are compounds which are not directly required for plant growth but are very important for plant survival. These include alkaloids, terpenoids, and phenolic compounds. Examples are morphine and flavonoids. These compounds are very important in pharmaceutical and industrial applications. They can be produced using plant tissue culture, suspension culture, or hairy root culture. Their production can be increased using elicitors and precursor feeding method. Hairy root culture is a special type of plant culture induced by Agrobacterium rhizogenes. This bacterium transfers genes into the plant, which cause formation of fast-growing roots called hairy roots. These roots grow without the need for external hormones. Hairy roots are very useful because they grow quickly, are genetically stable, and produce high amounts of secondary metabolites. That is why they are preferred over suspension cultures for metabolic production in many cases. In hairy root formation, the bacterium transfers a part of its DNA called RI plasmid into the plant cell. This DNA integrates into the plant genome and changes the normal growth pattern of the plant cells. Important genes called ROL genes are responsible for root formation. As a result, many roots are formed and metabolism is altered. For example, remember that this mechanism is similar to Agrobacterium tumefaciens, but the plasmid is different, RI plasmid instead of TI plasmid. Transgenic plants are plants in which a foreign gene is inserted artificially. This is done to introduce new traits like resistance or improved quality. The process includes gene isolation, vector construction, transformation, selection, and regeneration. Two main methods are used: agrobacterium-mediated transformation and gene gun method. After gene transfer, only the successful transformed cells are selected and then regenerated into whole plants using plant tissue culture technique. This method uses Agrobacterium tumefaciens, which naturally transfers DNA into plants. It contains a TI plasmid, which has a tDNA region that gets inserted into the plant genome. VIR genes help in this transfer process. The process involves infection of plant cells, transfer of tDNA, and integration into the genome. This method is widely used because it gives stable gene expression and usually inserts fewer copies of the gene. The gene gun method is also called biolistics. In this method, tiny particles of gold or tungsten are coated with DNA and shot into plant cells using high pressure. This allows DNA to enter directly into the cell. This method is mainly used for monocot plants like rice and maize, where agrobacterium is less efficient. However, it has some disadvantages like random gene insertion and multiple copies of gene, which can affect expression. Transgenic plants are widely used in agriculture. One important application is pest resistance, like Bt crops, which can kill insects, pests. Another application is herbicide resistance, which helps farmers control weeds easily. They are also used for improving nutritional value like golden rice, which has more vitamin A. Additionally, transgenic plants can be made tolerant to stress conditions like drought and salinity, which is important for agriculture. Plants produce many useful products which are important for industries. In pharmaceuticals, compounds like Taxol and vincristine are used as anti-cancer drugs. In food industry, plant products are used as flavors and colors. In cosmetics, essential oils are widely used. Other products include rubber and biofuels. Plant tissue culture has many advantages. It allows rapid clonal propagation, which means many identical plants can be produced quickly. It also helps in producing disease-free plants, which is very important in agriculture. Another advantage is that production can be happen throughout the year, independent of seasons. It also requires less space and helps in conserving rare and endangered plant species by growing them in laboratory conditions. Although plant tissue culture has many advantages, it also has some limitations. One major problem is high cost because it requires special equipment, sterile conditions, and skilled labor. Also, contamination by bacteria or fungi can easily spoil the culture. Another important limitation is somoclonal variation, where regenerated plants may show genetic changes. This means all plants are not always identical, which is a common exam trap. Some tissue culture is useful but not always perfect reliable. This slide is very important for last minute revision. It summarizes key concepts like totipotency, which means a single cell can form a whole plant, and the auxin to cytokinin ratio, which controls plant development. Also remember that secondary metabolites are mainly produced in the stationary phase. Another important plants include hairy root culture being best for metabolic production, agrobacterium being the most commonly used method for transformation, and MS medium being the standard culture medium. These are frequently asked direct questions in GAT-B. Subscribe Brainzz and stay updated with the latest GAT-B 2026 preparation.